Hey guys, Miss Cote here, and this is video 3 of the biochemistry unit. In this episode, we're going to look at the very abundant category of biological molecules called proteins. So let's get started. Proteins are extremely important in living organisms. They make up more than half of the dry mass of most cells, and they have such a wide variety of functions in the cell. Let's briefly go through them. Proteins can be enzymes, speeding up chemical reactions. And since enzymes are part of the biochemistry unit, we'll be looking at enzymes in more detail very soon. And we'll keep learning about different enzymes and their functions throughout the year. Proteins can help defend against bacteria and viruses. When we get to the immune system, probably in the spring, we'll look at the role of proteins called antibodies and how they neutralize foreign particles that get inside our bodies. They can be used for storage of amino acids, to be used as nourishment. Baby mammals can get amino acids from a protein called casein, which is the protein found in mother's milk. For animals that hatch from eggs, albumin, what you know as egg white, is the source of amino acids for the developing embryo. They can be used as a means of transporting substances. You guys should be familiar with the use of carrier proteins to move molecules across the cell membrane. We'll be discussing cell transport in more detail in the next unit. And during the course of the year, we'll talk about various molecules like hemoglobin, whose job it is to carry molecules such as oxygen around the body. Some proteins act as hormones, delivering messages throughout the body. This is the job of the endocrine system, one of the major body systems we'll discuss during the cell communication unit. Other proteins are receptors used in signal transduction pathways. Again, this will also fall into the cell communication unit. Some proteins are used for movement, such as actin and myosin, two proteins that are responsible for muscle contraction. And finally, proteins can be structural. For example, the protein keratin is the protein in hair. To sum all of this up, you're not going to get away from proteins because they're just everywhere. A human has tens of thousands of different proteins, and each one has a different three-dimensional structure. What's amazing is that all this diversity stems from the same set of 20 amino acids. As the amino acids are joined together, we call these chains polypeptides. It's the sequence or the order that these amino acids are joined together that makes a polypeptide unique. And then you can take this polypeptide or multiple polypeptides and join them together to make a functional protein. So let's talk about amino acids in more detail and see how the finished protein is made. An amino acid is the monomer or building block of a protein. Fair warning, you'll need to be able to recognize and identify this molecule. As you can see from the diagram, the elements it contains are carbon, hydrogen, oxygen, and nitrogen. The way that it's built is that you start with a central carbon atom, and it's covalently bonded to 4 different things. To the south, you have a hydrogen atom. There's an amino group to the west, our carboxyl group to the east, and up north, you have something called an R group. The R group is what differs between the 20 amino acids. An R group can be as simple as a hydrogen atom or more complex like a double ring structure. Just a note, while the amino group and carboxyl group are always on the left and the right, respectively, sometimes amino acids can be shown where the R group could be on the bottom and the hydrogen can be on the top, or vice versa, so just be aware of that. Here we have the 20 amino acids with the parts that are the same shaded in purple and the R groups shaded in other colors. The amino acids are grouped according to the properties of their R groups. The amino acids on top with the yellow R groups are nonpolar or hydrophobic amino acids. The R groups shaded in green are polar, which makes them hydrophilic. And on the bottom, we have the electrically charged R groups, which also makes them hydrophilic. As we shall soon see, the properties of the R groups will have an effect on the protein as a whole. And I know what you might be thinking, and don't freak out. You're not required to memorize the names and structures of these guys for this class. Just be able to recognize that it's an amino acid. Memorizing these is for a college-level biochemistry or organic chemistry class, in case that's something that you're interested in the future. Remember, to make a polymer, you need to complete a dehydration reaction. Two amino acids can be joined together by a covalent bond called a peptide bond. Here in the figure, the dehydration reaction is happening where water is being removed to make a polypeptide chain. Polypeptides can range from this tripeptide you see here, tri meaning 3 amino acids joined together, to a chain of over 1,000 amino acids. And since these chains are made from a set of 20 amino acids, each has a unique sequence with the amino end on the left and the carboxyl end on the right. Let me just say here, this polypeptide chain that is made from joining amino acids together is not necessarily a protein. To be functional, a protein can be one or more polypeptide chains that are twisted and folded into a three-dimensional shape. So let's take a look at how that happens. The function of a protein is due to its three-dimensional structure, and its three-dimensional structure begins with the sequence of amino acids in the polypeptide chain. The polypeptide chain then takes on its shape due to interactions between parts of the chain, and the shape it takes will determine its job. Remember, structure is related to function. All proteins share 3 levels of structure: primary, secondary, and tertiary. When a protein involves more than one polypeptide, then a fourth or quaternary structure happens. It's very important to understand the 4 levels of protein structure, especially the interactions at each level. College Board loves this stuff. So let's take some time to review the different levels. The primary structure of a protein is simply its sequence of amino acids. And if you remember from Honors Bio, this sequence is ultimately determined by a unit called a gene, which consists of DNA. And if you don't remember that, we'll be going over transcription and translation later on in the course. Secondary structure involves interactions within the backbone of the polypeptide. Hydrogen bonds start to form. Remember that oxygen atoms have a partial negative charge, and the hydrogen atoms attached to nitrogen have a partial positive charge. Therefore, hydrogen bonds can form between them. Depending on the number and location of the hydrogen bonds, 2 different structures can form from these interactions. The polypeptide can take on an alpha helix shape, which looks like a coil, or a beta pleated sheet shape, which resemble the folds of a fan. Note that both shapes can be found in a single polypeptide. The next level is tertiary structure. This is when the interactions between the R groups come into play. As the backbone forms either a alpha helix or the beta pleated sheet, the R groups from nearby amino acids can interact with each other. For example, 2 sulfurs near each other from sulfhydryl functional groups can form a disulfide bridge, which is a type of covalent bond. Ionic bonds can form between 2 charged R groups. Hydrogen bonds can form between R groups with partial charges on them. Remember, hydrogen bonds are represented by the 3 dots between the atoms. And nonpolar R groups can form hydrophobic interactions. Now, if the protein is made up of only one polypeptide, then that's all that needs to happen for this chain to become a functional protein. However, for a protein made up of multiple polypeptide chains, there is one more step. Quaternary structure involves multiple polypeptide chains coming together to form a larger functional protein. A classic example is the protein hemoglobin, which is made up of 4 polypeptide chains. two of one kind, called the alpha subunit, and two of another kind, called the beta subunit. I say classic example because your book likes to use hemoglobin and sickle cell disease as a model of what happens when a protein doesn't fold correctly. Actually, they like to use sickle cell disease to illustrate many concepts in this course, so you will hear about sickle cell again. Sickle cell disease is an inherited blood disorder where a mutation causes one amino acid in the polypeptide to change, and the result of that one change produces an abnormal beta subunit. This leads to clumping of the hemoglobin proteins, which changes the shape of the red blood cells and reduces hemoglobin's capacity to carry oxygen to the cells. Feel free to read up on the sickle cell example, but I'm not going to discuss it in this video. Kind of don't have time. So while the primary structure will determine the shape of the protein structure can also be influenced by other conditions, both physical and chemical. Changing the environment by changing pH, temperature, or salt concentration, for example, can cause the interactions at the quaternary, tertiary, and secondary level to be disrupted. This will cause a protein to unravel and lose its shape. We say the protein has denatured, and this denatured protein is no longer functional. Notice that in a denatured protein, the primary structure remains. Those covalent bonds, those peptide bonds, are very strong and can withstand any of the environmental changes. If the proper environment for the protein is restored, sometimes the protein is able to fold back to its original conformation, but know that this is not always the case. But how does protein folding actually happen in the cell? Even though scientists can figure out the sequence of a polypeptide, they're still having a difficult time predicting how that polypeptide will fold. Protein folding appears to be a complicated process that may occur in various stages. There's actually an online game called Fold It, where you're given the primary structure of a protein and your job is to figure out how it could potentially fold. Scientists actually look at the solutions and see if they actually apply to proteins in real life. Helping the protein fold into the correct conformation requires another protein called a chaperonin, and it's also called a chaperone protein. Looking at the diagram, the chaperonin kind of looks like a thermos. The body has a hollow cylinder with a cap. How it works is that a polypeptide goes in, the cap is closed, and the polypeptide is allowed to fold under optimal conditions. Then it's released back into the cytoplasm to do what it needs to do. I know this was a bit of a long video, but proteins are pretty important, and there's a lot you need to know about them. The next episode in the biochemistry series will be nucleic acids. And if you have any questions, please feel free to bring them up in class. So until next time, let's be good humans.